Thin layer chromatography, TLC, is used for the separation of nonpolar and slightly polar molecules. This technique is used to monitor organic reactions, identify compounds, test for purity, prepare solvent systems for column chromatography. It is quick, cheap, and requires only a small amount of analyte. TLC incorporates using a solid phase, the silica, a polar compound. The mobile phase is the solvent system, and the interactions between molecules, your silica, and the mobile phase leads to separation of the molecules. This separation can be visualized as spots and the RF value calculated and an unknown identified. Today, the task is to identify one unknown compound using several mobile phases. Using a soft pencil, gently draw a baseline 1 cm from the bottom of the plate. Do not scratch or damage the silica surface. Do not lay the ruler on top of the main area of the plate. Lightly mark 4 equidistant positions, at least 1 cm away from the edge of the plate. Label each position with a letter to identify your known and your unknown compounds. Also identify in the top right-hand corner the solvent system of your mobile phase. Complete all 4 plates in the same manner. Using a microcapillary tube, spot your compound onto the plate. The tube is fragile, so please do handle with care. Place the capillary tube into your first compound. Spot gently and quickly onto the marked position on each plate. Small spots are required for good TLC result. If necessary, wipe or dab the end of your capillary tube with tissue to remove any excess drop of your compound. Wash the capillary tube 3 times in ethanol to prevent cross-contamination. Repeat the spotting and washing technique for your remaining compounds. Using the tweezers provided, place the plates upright in the correct mobile phase. Do not let the plate fall backwards or forwards, and do not move the mobile phase bottles once the plate has been placed inside, as this could ruin the spot and the RF value. Observe the mobile phase rising up the plate, and when it is 1 cm from the top of the plate, remove it using the tweezers. Draw a pencil line very gently at the top of the solvent front. Replace the lids on the mobile phase and allow the solvent to evaporate from the plate. Using a UV lamp, visualize the spots. Caution: do not direct the light into the eyes. Hold the light above the TLC plate and draw gently around each spot. You may now determine the RF values of your compound and identify your unknown. On completion of the practical, the TLC plates must be placed in the correct waste container provided. Dispose of the mobile phases in the appropriate waste bottles in the fume hood.
This video outlines a method for carrying out column chromatography. You may be given slightly different instructions when you do this in the lab, but the principles will be the same. First of all, a small piece of cotton wool is placed in the bottom of the column to prevent solid material from going into the tap. Different columns may have different types of tap, but obviously the idea will be the same. Next, a small amount of sand is poured in to a depth of about 1 centimetre. Here you can see the sand forming a level surface on top of the cotton wool. We then take the appropriate amount of silica in a beaker, and this is then made into a slurry using the solvent we're going to run the column with. This solvent is known as the eluent. Here you can see us swirling the silica with the solvent before pouring it into the column very carefully. Again, a funnel should be placed in the neck of the column to assist with pouring. And you will need to occasionally swirl the beaker to ensure that the silica doesn't settle out in the bottom of the beaker. Where appropriate, more eluent should be added to reform the slurry to assist with pouring. You may need to do this several times. Should the level of the eluent get close to the top of the column, then some of it should be removed by opening the tap. We can run this into the beaker, which will then assist us in making a slurry with the remaining silica. And finally, we've transferred all of our silica into the column. Now we need to run some of the eluent out of the column so that it comes down to the level of the silica. It's crucial that you don't allow the silica to run dry, so you need to keep a close eye on the level of the eluent and close the tap at the appropriate moment. Here you can see we've run the eluent down to the correct level so that the top of the silica has not dried out. The next stage is to load our crude compound onto the column. First of all, we need to dissolve it in a small amount of the eluent. It's important to ensure that all of the crude compound has been dissolved. And then the resulting solution can be added to the column using a glass pipette. Here you can see the solution is being added evenly around the sides of the column. Now we add some sand to a depth of about 1 centimetre on the top of the column. This will protect the silica as we add eluent. And here you can see us topping up the column with eluent. And we're now ready to run our column. So we open the tap at the bottom of the column, and this allows us to collect our first fraction. In this case, we're collecting the fractions in test tubes, but depending on the size of your column, you may use sample vials or even conical flasks. Once collection is complete, we can close the tap momentarily to move on to collect fraction number 2. And this process can be repeated any number of times to collect as many fractions as are required. Because our compound is coloured in this case, we can actually see it moving down the silica. This will give us a good indication as to when it's actually eluting from the column. In cases where you can't see the compound because of any colour, you can use TLC to analyse the fractions to identify the presence of different compounds. And as the level of the eluent approaches the sand, it's important to make sure that you top it up. We can then continue to collect fractions, and in this case you can see that the yellow band in the silica is approaching the bottom of the column. If we look closely at this particular fraction, we can see that the solvent has a yellow colour, telling us that our compound is eluting. Of course, in other cases, as mentioned previously, you'd have to use TLC to identify the presence of a compound. And here we have a full set of fractions. We can now collect any compounds that are present by removing solvent, and this would most commonly be done using a rotary evaporator.
Paper chromatography. Separate the colored components present in the mixture of red and blue inks by paper chromatography. We require a mixture of red and blue inks, distilled water, isopropyl alcohol, Whatman filter paper strip, pencil, scale, capillary tube, chromatographic chamber, etc. Take a Whatman filter paper strip. Draw a line with the pencil above 4 cm from one end. Draw another line lengthwise from the center of the paper and name the point at which the two lines intersect as P. Take a capillary tube. Dip it into the beaker containing the mixture of red and blue inks and take the mixture using the capillary tube. Put a drop of the mixture of red and blue inks at the point P. Allow it to dry in open air. Again take the mixture of red and blue inks using the capillary tube. Put another drop on the same spot and dry it again so the spot is rich in mixture. Measure 5 ml of distilled water in a measuring jar. Pour it into the chromatographic chamber. Measure 5 ml of alcohol in another measuring jar. Put it into the chromatographic chamber containing distilled water. Mix the contents of the chromatographic chamber using a glass rod. This mixture is used as the solvent. Suspend the filter paper vertically in the chromatographic chamber containing the solvent in such a way that the pencil line remains about 2 cm above the solvent level. Close the chamber with its lid and keep it undisturbed for some time. Notice the rise in solvent along with the red and blue inks. When the solvent has risen to about 15 cm, you will notice 2 different spots of red and blue inks on the filter paper. Take the filter paper out of the chamber and mark the distance that the solvent has risen on the paper with a pencil. This is called the solvent front. Dry the filter paper in open air. Put pencil marks in the center of blue and red spots. Measure the distance of the solvent from the original line and the distance of the 2 spots from the original line. Calculate the RF values of blue and red inks by using the formula. RF is equal to distance travelled by the solute from the original line divided by distance travelled by the solvent from the original line. Separation of pigments from the extract of spinach leaves by paper chromatography. We require extract of spinach leaves, distilled water, isopropyl alcohol, Whatman filter paper strip, pencil, scale, capillary tube, chromatographic chamber, etc. Take a Whatman filter paper strip. Using a pencil, draw a line that is above 4 cm from one end. Draw another line lengthwise from the center of the paper and name the point at which the two lines intersect as P. Take a capillary tube. Dip it into the beaker containing the extract of spinach leaves and take the mixture using the capillary tube. Put a drop of the spinach leaf extract at the point P and allow it to dry in open air. Again, take the extract of spinach leaves using the capillary tube. Put another drop on the same spot and dry it again so that the spot is rich in mixture. Measure 5 ml of distilled water in a measuring jar. Pour it into the chromatographic chamber. Measure 5 ml of alcohol in another measuring jar. Pour it into the chromatographic chamber containing distilled water. Mix the contents of the chromatographic chamber using a glass rod. This mixture is used as the solvent. Suspend the filter paper vertically in the chromatographic chamber containing solvent in such a way that the pencil line remains about 2 cm above the solvent level. Close the chamber with its lid and keep it undisturbed for some time. Notice the rise in solvent along with the leaf extract. When the solvent has risen to about 15 centimeters, you will notice different spots of the components of spinach leaf extract on the filter paper. Take the filter paper out of the chamber and mark the distance that the solvent has risen on the paper with a pencil. This is called the solvent front. Dry the filter paper in open air. Put pencil marks in the center of each spot. Measure the distance of the solvent front from the original line and the distance of the spots from the original line. Calculate the RF values of the different spots by using the formula. RF is equal to distance travelled by the solute from the original line divided by distance travelled by the solvent from the original line.